So one of the things that we've identified early on in the piece was what can we do to push microbes in the right direction using new products or using new strategies. So if you sort of consider what happens in a forest to keep it going, we have material sitting in organic matter, resources in plant biomass. And generally we think we've got resources in the soil, they get converted into the plant biomass, that stuff dies, falls off, whatever, it hits the soil again and becomes organic matter that eventually becomes available for use again as a resource. However, in a lot of our forest soils, what we see is a build-up in that organic matter phase, and that's why we can have a lot of resources sitting in a site, but we just can't use them, and that's some of the problems that we have at our accelerator trials. So we became interested in the use of this molecule called Biuret as an opportunity to increase the breakdown of that organic matter and to get those resources back into the cycle and to upregulate this whole process. Now, why did we play with Biuret, and what actually is it for starters? Well, Biuret is something you used to get when you make urea badly. It's essentially a dimer of urea, two molecules stuck together. It's got a lot of nitrogen in it, but it's about 50 times less soluble than urea. Now, early work done with Biuret by Simeon showed that it did have positive effects on microbial function and activity, and that's why we got really interested in it. However, Biuret's also a really potent phytotoxin, which is less good. So we did see some research showing that Douglas fir had some level of resistance to the negative effects of Biuret, so we thought, well, what can we do with Pinus radiata? Can we find a way to use it? So our initial work focused on trying to find a sweet spot where the benefits that you got from increasing the microbial activity outweighed any losses we might see from toxicity. So we threw it in a scientific way at a lot of really just recently emerged seedlings because we figured they'd be about as sensitive as you could get. However, we found that what should have been a mortality trial ended up being a growth promotion trial. We just couldn't get any toxic effects. And we added more and more and more, and simply the more Biuret we added, the faster and the better the seedlings grew. And this lack of toxicity opened the door to a whole suite of experiments and trials that we've conducted since, because all we saw were positive results in a number of ways. And one of the first positives we saw, was increased uptake efficiency of nitrogen. So on this axis here, you've got the control, then you've got 4 increasing levels of Biuret compared to 2 increasing levels of urea. There was a third one, but it was too much and it killed the seedlings stone dead in about 12 hours. But what we saw is that the more Biuret we added, the more efficiently it went into the tree. And again, we've seen this consistently played out in a number of other trials as well. But it goes beyond the uptake of nitrogen. We've taken this quite rapidly to operational scale trials in the nursery. That F100 treatment there, that is the standard conventional dose of nitrogen, 100%, delivered as conventional fertilizers. We compared that to a 25% dose of nitrogen and a 50% dose of nitrogen added in the form of Biuret. Now, not only did the trees getting less nitrogen, but in the form of Biuret grow faster, we also found that they increased their uptake of nitrogen. So actually they had more nitrogen in them, but they also picked up more boron and they also picked up more potassium. We didn't change those, but what we found was basically the system was working to deliver more to the trees. We were adding less, they were getting more. And this is where we started taking it out into the field. So again, you heard Loretta mention this 500 kilogram gap of nitrogen we need to get into the trees. We need to get into that organic cycle to make the forest hit our productivity targets. So we thought, well, what can we do with Biuret to stimulate microbes to increase access to the nitrogen that's already there? So we added about 12.5 kilograms of nitrogen per hectare in 2 doses to the accelerated trial, and we very rapidly saw significant growth gains, and the uptake far surpassing what could possibly have been delivered by our treatment. So we think we're actually on track to start stimulating the microbes in a real-world example and to put ourselves on the better growth trajectory. The other opportunity we took with Biuret is to take that stimulation of microbial activity and see what we could do with ACC deaminase, which Sarah just talked about. So again, exploring the variation with genotype out in the field, we saw huge variation. We had some clones down here, sort of in almost single-digit gene copy numbers, up to things in the thousands. And again, we've got some of our high-performing, our very resistant clones are naturally really good at attracting the bacteria that help them with that resistance. So we thought, well, what can we do about these guys, though? These ones might have some really interesting wood properties. We want to see if we can make them more tolerant, so what can we do here? So we added Biuret, And lo and behold, we found we got some really significant gains in the gene copy number of ACC deaminase, but just in response to that small dose of Biuret. However, we did find that there are some genotypes that were already pretty good at recruiting ACC deaminase-containing bacteria. The best course is just to leave them alone. Whatever happened, we possibly overstimulated that association. But either way, we've got a tool now, a new tool, to increase the stress tolerance of less tolerant clones that might have properties we want otherwise. And I'd like to say that even despite some of the trees responding in this aspect negatively to Biuret, overall everything responded really positively. So we still saw significant growth gains across the board. Another aspect we're trying to use is the actual phytotoxicity of Biuret. Can we actually make that work for us? So an example of some of the research we've been doing, what we did is we took a number of seeds of various weed species that are problems in the nursery. We put them in soil, we stratified them up, put them in soil, and then treated that soil with Biuret. Now, initially, we thought our experiment was a bit of a failure because what we saw is that everything germinated. The ones that had higher Biuret took a little bit longer to germinate, but they still got germinated. But then when we ran the experiment for a little bit longer, what we saw is that the seeds that had germinated and were growing in soil treated with a higher rate of Biuret, they were dying before they could reproduce. So in the control, there we go. Oh, that'll do. In the control plot, we had thousands of progeny of our initial weeds, hundreds, tens, single digits. And this was seen over a matter of a couple of months. So this really interested our nursery staff. Again, totally unexpected, but still another very promising result. And I suppose lastly, the other thing that we've seen with Biuret is we've basically at the Southern Kaingaroa Accelerator trial, we've linked Biuret application with increased tolerance to Dothostroma. So what we have here is that the trees that were treated with Biuret showed less damage from Dothostroma. We don't think this is a direct effect whatsoever. It's just simply the trees are healthier. We think they're better off and better able to resist the disease on their own terms. Now, based on all of these outcomes, we are actively working to try and make Biuret commercially available in New Zealand. So this sort of leads us back to where we started. We've got all these opportunities to stack up productivity, and we're pursuing all these opportunities in the accelerator trials. However, there's another thing that we're using the accelerator trials for as well, and that's to look at the uncertainty that's going to be facing the forestry sector in the next 25 years, the next 50 years due to climate change. There's a real possibility that some of the targets we set for ourselves because of changes in climate, we will have a really hard time reaching those simply because of negative environmental effects that aren't manifest right now. So we're trying to figure out how we can build as much resilience into our accelerator trials so that whatever happens to the climate, we can still hit those productivity gains. We can still be on track. And on the other side of that is that climate in some places will actually increase the potential growth. The potential yield may go up. So again, within the accelerator trials, we're trying to build as much adaptability into our framework so we can take advantage of good conditions. We can look to do more with what's at a given site and hit maximum productivity, whatever it is now or potentially in future. And that's basically what the research we're doing. We're looking to enhance productivity at the site now, improve the crop now, but improve the crop for the future and the site for the future.